Warm greetings to everyone. I am Winnie Wilson. I'm a third MSc Biotechnology student who's studying in Christy Jayanti College, Bangalore. And today I'm going to talk about enzymatic method of DNA sequencing, which is guided by Dr. Manikandan. These are my contents, and my presentation will be included of introduction, principle, procedure, applications, conclusion, and references. Introduction. Enzymatic tool of DNA sequencing was the first virtual tool to sequence large DNA. It is also known as polymerase method due to the usage of DNA polymerase. This method is based on Sanger's work, in which a description of Sanger's DNA sequencing by using the chain termination inhibitors known as dideoxy method is being used. Sequencing method is based on template direction and the primed synthesis of single-stranded DNA. Along with Maxim Gilbert method and using DNA polymerase, this method provides a reliable and simple way for DNA sequencing. Based on the ability of extending primer by DNA polymerase after hybridization to template to be extended till the chain termination nucleotide gets incorporated. To terminate the single-stranded DNA in the sequencing process, DNA fragment to be sequenced binds with the primer. So this is the overall reaction. So the principle. So for as a short, the single-stranded DNA it undergoes a enzymatic synthesis of complementary polynucleotide chains, which then undergoes termination at specific nucleotide positions. Then it is separated by gel electrophoresis and the DNA sequences are read. Principle of enzymatic sequencing. DDNTPs are used as chain terminators of DNA, which is the key principle of enzymatic method of DNA sequencing. The following are required for a chain termination method. Those are single-stranded DNA template, DNA primer, DNA polymerase, fluorescently labeled nucleotides, and modified nucleotides that can terminate DNA strand elongation. The DNA sample is divided into four separate sequencing reactions containing all four standard deoxynucleotides, that is DATP, DGTP, DCTP, and DTTP, and DNA polymerase. One of the four D-deoxynucleotides are each reaction. The chain terminating nucleotides that lack 3-OH group needed to form a phosphodiester bond, leading to the termination of DNA strand extension, and the results in varying of DNA fragments. The new DNA fragments, which are labeled and synthesized, are then denatured by heating. They are separated by size with a resolution of one nucleotide by the process of gel electrophoresis. The apparatus contains denaturing polyacrylamide urea gel. Each of the four reactions are run in individual lanes. The DNA bands are then visualized by autoradiography or UV light. The DNA sequences can be read directly off the gel image or X-ray film. When the X-ray film is exposed to the gel, dark bands corresponding to DNA fragments of different lengths. A dark band indicates one DNA fragment as a result of chain termination. So this is obtained after the incorporation of any deoxynucleotide. So for the procedure for the entire process of enzymatic method, first the template DNA is being chosen. So the DNA to be sequenced is the template DNA. So for its preparation, single-stranded DNA is spliced into M13 vector DNA. The infected host cells of E. coli release phage particles containing a single-stranded recombinant DNA, which includes the sample DNA. The DNA sample is extracted from phages for sequencing purpose. Use of a primer. synthetic 5' end labeled oligonucleotide is used as a primer here. Hybridization. Template DNA is then hybridized to the primer. Fourth step, elongation. Elongation of the primer is done separately in 4 polymerization reaction mixtures. Each reaction mixture has 4 normal DNTPs in higher concentrations and each of the 4 DDNTPs at lower concentrations. Fifth step, initiation. DNA synthesis is then initiated by adding DNA polymerase as the enzyme cannot distinguish between normal nucleotides and analogues based on them. The next step, continuation of elongation. Strand synthesis continues till DDNTP gets added. By incorporation of DDNTP, the elongation stops. This happens because it lacks a 3'OH group, which helps in preventing the addition of the next nucleotide. The next step, obtaining mixture. A mixture of terminated fragments of different lengths are obtained. Denaturation. DNA fragments of the mixture are then subjected for denaturation, which is followed by gel electrophoresis, as these mixtures are run on a polyacrylamide gel. The next step is autoradiography. The separated DNA fragments from the mixture are visualized through autoradiography, and by radiogram, the position of the bands and the sequence of original DNA template strand can be directly read. So, this is the overall reaction which I've just summarized. The applications of enzymatic method. First one, CRISPR and TALEN mediated genome editing. The enzymatic method of DNA sequencing can be used for determining the accuracy of clustered regularly interspaced short palindromic repeats and transcription activator like effector nucleases mediated genome editing techniques in organisms. The second applications for SARS-2 research. Sequencing technique can be used for complementing laboratory techniques that can lead to efficient research of SARS-CoV-2. The third application, next generation sequencing. The enzymatic method can be used as an orthogonal method for confirming variants which are identified by the next generation sequencing using ion amplisec primers and libraries. The technology of ion amplisec is ideal in routine labs by working with a limited number of samples and high throughput laboratories that need an orthogonal method to confirm minor alleles. This next application, detection of low level somatic variant in tumor FFPE samples. This enzymatic method can be used ideally for low level variance detection in the material containing small amounts of DNA like formalin fixed paraffin embedded tissues. The next application, mitochondrial sequencing. Useful tool to study human diseases like diabetes, cancer and mechanism of aging. It can also be used in the fields of population genetics, biodiversity assessments, human identification, and forensic applications. The last application is HLA typing. Human leukocyte antigen, otherwise known as HLA, typing can be used for matching patients and donors for bone marrow and cord blood transplants. As a conclusion, the enzymatic method of DNA sequencing uses single-stranded DNA. Known as D-deoxysequencing method, this technique includes the use of analog of normal nucleotide of DDNTPs. The technique is then based on the incorporation of DDNTPs in a growing DNA strand for stopping chain elongation. By adding DNA polymerase, the primer is extended till a DDNTP encounters. By the proper DNTP-DDNTP ratio, the chain terminates throughout the length of the template. The collection of fragments forms a sequencing ladder. Terminated chains are then resolved through electrophoresis. So this is the overall reaction once more. And these are my references. Thanks for watching and listening to this presentation.